Hello everybody, my name is Bruno Folchi from AgroVentures, a startup company developing ecological crop protection solutions. I'd like to update you on the progress of our project to reduce impact of chemical nematicides by using a nematostatic wild yeast strain. We call it YSY Yeast, and it's based on Papiliotrema terrestris. We introduced this concept during last July IR4 technology session. We presented the summaries of a recent publication from a team of Italian universities that tested Papiliotrema terrestris for nematode control on strawberry. Nematodes, in that case, were Meloidogene species. Multiple applications, in this case about 6, led to a control around 50 to 60% comparable to the chemical reference tested, fluopiram in this case. Mode of action was identified as a fast reduction of nematodes mobility, comparable to that provided by products like phosphoazate and abamectin based. Now I'd like to update you about further confirmatory trials we performed in 2024. Meloidogene control on tomato was good again, and these are the average results from 8 trials in Italy. Again, we confirm around 50% control plus, comparable to fluopiram and to the best alternative biocontrol options. Interestingly, IPM program can work well by maximizing performance of both chemicals and our yeast biocontrol. Same results were noticed on strawberry. Basically, the mode of action is the same across multiple crops. We optimized the protocol by reducing a number of applications, so we think we can really be a competitive option, considering rate is only 1 kilo per hectare or 1 pound per acre. Nematode control is not practiced in isolation, but always linked to other plant protection and soil fertilization programs. Therefore, we are evaluating easy complementary action on other main factors affecting plant health. For instance, soil-borne disease control. Here we have good results. In fact, YSY Yeast was born as a biological fungicide, and we have consistent data showing soil-borne disease controls like Rhizoctonia, Pythium on multiple crops. Here you can visualize the average results from our trials on tomato in Italy last year on Pythium only. So 50% plus control comparable to trichoderma and the chemical reference, phosphatil, propamocarb in this case. Focusing on soil ecology, we also perform metagenomic studies on multiple soil types to understand how YSY Yeast modifies the microbiome, especially beneficial organisms, as those involved in nematode control. In general, YSY reduces the amount of pathogenic microorganisms while not significantly affecting beneficials. And these findings are generating interest for nematode bioconsortia evaluations. We are testing various combinations with research stations, and results are planned for publication this year. On the table you can see basically the evolution of different microorganisms in the soil media, and again all those positively affecting nematode control don't have any significant change. We're also studying how our yeast contributes to soil and plant health with encouraging results on biostimulation. We have multiple data on tomato, melon and other vegetable crops. We just give you an idea of relative performance on lettuce when applied on soil. So when you apply to control nematodes or plant diseases, these are results that are quite common on the main vegetative parameters. So there is a significant growth or number of leaves, dry weight of the roots and the aerial part, to level comparable of specific root stimulants like Radifarm based on combination of vitamins, amino acids and other micronutrients. With this data, we are also starting to test YSY Yeast activity as a seed treatment with initial positive findings. And this is the data for a test we had on corn, where again all the main vegetative parameters emergence to begin, and then vegetative parameters are improved. Biostimulation is important and in our case is even more noticeable under abiotic stress conditions, for instance on salty soils. This is data from another recent publication by a consortium of Italian research stations, where basically YSY Yeast proved to reduce mortality by salt stress significantly and close to the best known other available options. And you can also visualize how severe the damage for salt stress was under testing conditions and how YSY Yeast contributed to mitigate that damage. Last but not least, we are investigating YSY potential for environmental and human health protection. And as expected, the conclusion is that YSY use may dramatically contribute to chemicals impact reduction. In this slide, we summarize the summary from a life cycle analysis study that we will also publish this year, where you can see that for multiple impact categories, YSY Yeast can reduce impact from chemical fumigants by tens, hundreds, thousands of times. You can see here, for instance, for carbon footprint reduction, we could reduce by 320 times the impact of methane sodium, 12,000 times ozone depletion for chlorpicrin, and so on. Data is quite dramatic. All this evidence increases our determination to develop a nematode control solution with additional benefits that combined can add significant value to plant health and environmental protection programs. And we are working to register YSY Yeast in Europe and the US, hopefully in the next couple of years. More information on the product can be found on the links below. Thank you very much.